All right, let's jump straight into this explainer. If you're tackling higher-level biology, you already know the deal. During the S phase of the cell cycle, a cell has to duplicate its entire genome before it can even think about dividing. But today, we're not just looking at this as some abstract biological concept. We're going to treat it like what it really is, a highly coordinated, high-speed molecular assembly line.
Here's a quick look at our agenda for today. We'll start with the master blueprint, move into initiation to open the helix, hit the assembly line and elongation, check for errors during termination and quality control, and finally, look at eukaryotic complexity.
Okay, part 1. The master blueprint.
To really get genetic inheritance, you have to lock in this core rule, semi-conservative replication. Just look at how brilliant this is. Every single time a cell divides, it keeps exactly half of the original structure. It literally unzips the double helix right down the middle, and each old strand becomes a flawless template for a new one. This guarantees absolute perfect fidelity across generations.
But, that immediately brings up a huge logistical headache. How on earth does the cell actually unzip that blueprint? It's a massive, tightly wound double helix. So how do the molecular machines even get inside to start copying?
Well, let's look at Section 2, Initiation, Opening the Helix.
Let's step onto the factory floor, using our good old friend E. coli as the baseline. It all kicks off at one highly specific address called ORI-C. An initiator protein, DNA-A, binds there. Now, because this spot is packed with adenine and thymine, which, remember, only share 2 hydrogen bonds instead of 3, it's just way easier to melt apart. So DNA creates this tiny starting bubble. That gives our motor protein, DNA-B, or helicase, the room it needs to barge in and aggressively unzip the fork. And to keep those freshly separated strands from instantly snapping back together, single-strand binding proteins, or SSBs, swoop in to coat them.
Now imagine grabbing 2 intertwined ropes and pulling them apart in the middle. What happens? The unpulled ends get incredibly tight, right? DNA does the exact same thing. Unzipping it creates massive torsional strain downstream. Enter topoisomerase, specifically called DNA gyrase in prokaryotes. This thing is the ultimate stress reliever. It carefully snips the DNA, lets the supercoil relax, and seals it right back up ahead of the replication fork.
Moving to Section 3, Elongation, the assembly line.
All right, with the helix safely open, the real work begins. But there's a huge catch. DNA strands are anti-parallel, and new synthesis can only, and I mean absolutely only, happen in the 5-3 direction. So, the leading strand? It's got it incredibly easy. It naturally runs 3-5, so the new strand just continuously builds 5-3, happily chasing the replication fork as it opens. But the lagging strand? Oh man, the lagging strand is a constant headache for the replisome. It's facing the completely wrong way. It has to be built backward, in short, discontinuous chunks moving away from the opening fork.
To handle that structural chaos, the assembly line relies on a strict step-by-step checklist. First off, DNA polymerases can't just start from nothing. They need a primer. So primase drops down a short RNA primer to give a starting 3' OH group. Then DNA polymerase 3, the absolute heavy lifters here, jumps on and builds a short stretch of DNA. We call that an Okazaki fragment. But wait, we can't just leave chunks of RNA mixed into our DNA, right? So DNA polymerase I swoops in, chews up that RNA primer, and replaces it with proper DNA nucleotides. Finally, DNA ligase acts as the chemical glue to seal the gaps in the backbone.
Section 4, Termination and Quality Control. Our little E. coli factory is blasting along at a blistering speed of about 1,000 nucleotides per second. At that speed, mistakes are totally inevitable. But check out this final error rate, 1 in a billion. Literally, 1 in 10 to the 9th. The polymerase actually makes a mistake about once every 10 to 100,000 bases initially, but the proofreading enzymes are so remarkably good that the final rate drops to 1 in a billion. That's like retyping an entire encyclopedia set and making exactly one typo.
So how do they do it? Well, polymerase 3 isn't just a builder, it's an inspector. If it slaps down the wrong base, its 3-5 exonuclease activity kicks in. Think of it like a biological backspace key. It steps back, cuts out the mistake, and tries again. And for shutting the whole factory down? In circular bacterial DNA, there are specific TER sequences right opposite the origin. TUS proteins park themselves there, acting as a literal physical stop sign to block the helicase when the 2 forks finally crash into each other.
Finally, Section 5, the eukaryotic complexity.
Let's scale this whole process up from a simple bacterial loop
to something massive, like humans. Eukaryotic replication is actually way slower, only 50 to 100 nucleotides a second. Why? Because our machinery constantly has to navigate around nucleosomes, dismantling and reassembling those histone proteins that pack our DNA tightly. To make up for being so slow, human cells fire off up to 100,000 origins of replication all at once. We also swap out parts. We use a different sliding clamp called pCNA to keep things on track, and enzymes like RNase H and FEN1 to clean up the primers.
Plus, our workforce is highly specialized. We don't just rely mostly on one Pol3 enzyme. Eukaryotes split the labor across an absolute alphabet soup of Greek letter polymerases. Polymerase alpha starts the job by laying down the initial primer. Then it hands the baton over to polymerase epsilon to sprint down the continuous leading strand, while polymerase delta takes over to handle all those complex stop-and-go Okazaki fragments on the lagging strand.
But because our chromosomes are linear, we hit a massive mechanical wall. What happens when that lagging strand reaches the absolute end of the line? Because eukaryotic chromosomes aren't closed circles, we face a critical flaw known as the end replication problem.
Here's the issue. When that final RNA primer is ripped out at the very tip of the chromosome, there is zero upstream DNA for the polymerase to grab onto to fill the gap. Without a fix, our chromosomes would literally just get shorter every time a cell divides, eventually eating right into vital genes. Enter telomerase. This enzyme is a total game changer. It actually carries its own built-in RNA template and artificially extends the ends of the chromosome with protective repetitive sequences called telomerase. That gives the standard machinery just enough runway to lay down one last primer and finish the job without losing any critical data. So, as you think about the unbelievable precision of this molecular assembly line, ask yourself this. Telomerase is completely turned off in most of our adult somatic cells. How does the gradual shrinking of those telomeres dictate our biological aging and mortality? And on the flip side, what happens when telomerase dangerously overactivates and fuels the unchecked cellular division we see in cancer? It's a razor-thin line between the blueprint of life and the mechanisms of disease. Thanks for joining me on this explainer. Keep asking questions and keep exploring the incredible machinery right inside you.